We want to think about the nature of the ultrafiltration process, that is the actual formation of the glomerular filtrate. Now, as we probably already know, we have the afferent arteriole and we have the efferent arteriole, and between those there is the ball of capillaries called the glomerulus, which is here. And it's a ball of capillaries, lots of complicated little capillaries. I've just drawn a few in there. And round about that we have the Bowman's capsule, or the glomerular capsule, going into the proximal part of the convoluted tubule. So the blood's coming in here, circulating through these capillaries, and draining into the efferent arteriole. So these are all the capillaries in here. The glomerular capillaries. This ball of capillaries, which is the glomerulus. And the glomerular filtrate is forming. It's being filtered by this process of ultrafiltration, and it's going into the glomerular space here, draining down through the glomerular space into the proximal part of the tubule. This area will soon become the proximal convoluted tubule. So that's what's happening in basic terms. Now, what's actually happening with a little more detail? Well, if we think about this capsule round here, this is the parietal part of Bowman's capsule. So here we have the basement membrane, and there are parietal cells on that basement membrane. So these are the parietal cells of Bowman's capsule, each with their own nucleus cytoplasm. So this wall I've just drawn here is like one of these outer walls of Bowman's capsule, and that is parietal. That is the outer membrane of Bowman's capsule. Now I'm just going to concentrate on one capillary. So I'm going to draw one capillary here, and that is the cross-section of just one of the capillaries in the glomerulus. And there's going to be a basement membrane around about there like that. In here, this is the lumen of one of the glomerular capillaries. Now, as you would expect, inside this basement membrane, there is the vascular endothelial cells. And it is the same as any other blood vessel, but the vascular endothelial cells lining the glomerular capillary have very large gaps in their cytoplasms. So there's large gaps. So instead of the cell being fairly solid, there's like gaps in between bits of the cell. So there's like little gaps in between components of the cell. So diagrammatically we could draw this something like this. These are the vascular endothelial cells. Vascular endo. The vascular endothelial cells. And they're going round the inside as you would expect. But what we notice is that there are relatively large gaps. So not all of these are individual cells. Some of these are just like finger projections from cells. So you might just see the odd nucleus. And these large gaps are quite deliberate. They're called fenestrations. Fenestrations. So here we have a fenestration. Here we have a fenestration. And fenestration is just a word that means window. So there's plenty of space for the process of ultrafiltration to take place from the blood in the lumen of the glomerular capillary through into Bowman's space over here. But there's more to it than this. Surrounding each capillary, there is a layer of specialized cells called podocytes. So here, for example, we have a podocyte. And the podocytes send small foot-like projections called pedicles down. The pedicles go down onto near the surface of the glomerular capillary. So you get something that looks a bit like this. These are the pedicles on the surface of the basement membrane. And of course, when you go around a bit more, there's going to be another one just here. So these pedicles are wrapping around the basement membrane of the vascular capillaries. And what this means is the small gaps between these cells, not between the cells, between the pedicles, between the foot-like processes. So these large cells here are the podocytes, and these pedicles are their foot-like processes. And these form the visceral layer. So the parietal layer is the outside of Bowman's capsule. The visceral layer of the glomerular capillaries surrounds each individual capillary. And the small slits here between these small foot-like pedicles. And actually, in these small slits, there's actually a slit membrane, an additional membrane. So what this means is, for ultrafiltration to take place, the fluid must come from the lumen of the glomerulus, through the fenestration, through the basement membrane, the glomerular membrane, through the slit process, then it has to fit between two pedicles of the podocyte, and only then does the fluid get into here, and this is the glomerular space. So the fluid's going from here, through a fenestration, through a slit membrane, into the space, into the glomerular space. And from the glomerular space, the fluid will go down and on towards the proximal tubule. And despite all this complexity, the distance here, the distance of the actual dialyzing ultrafiltration membrane, is only about 1 micrometer. So the distance that the glomerular filtrate has to travel from the lumen of the capillary into the glomerular space is only about 1 micrometer. Very small, but yet performing this ultrafiltration process, because the material must fit through the fenestration, go through the glomerular basement membrane, and fit between two pedicles. And the gap between any of these two pedicles with the slit membrane is only about 40 nanometers. That's only a 40 nanometer gap, tiny little gap in between. So that's just an introduction into the nature of the formation of glomerular filtrate. In the next clip, we'll take a look at a couple more diagrams that explain the way the pedicles and the podocytes interact with the glomerular capillary.